Hello everyone, my name is BeAnn and I am the main author of this study entitled Ganoderma lucidum treated oil palm empty fruit punch, a potential feedstuff for goats. So why did my study choose oil palm empty fruit punch as a potential feedstuff for ruminants? Well, firstly, oil palm is not only abundantly available, but it is also economically productive for 25 to 30 years with minimal maintenance. Secondly, EFB also has the highest percentage of waste generated annually in palm oil mills, yet it is still underutilized. Well, it is, however, a lignocellulosic fiber raw material, which means that it has more fiber than other nutrients, and this material can be easily digested by ruminants. Thus, in order to combat that, biological pretreatment of EFB was used. So, based on previous studies, biological pretreatment using white rot fungi has been used through solid-state fermentation, and this fungi is believed to be the most effective lignin-degrading microbes in nature. A study by Nur Nazratul et al. Back in 2019 has also used EFB treated with Ganoderma lucidum with 12 weeks of incubation, and the study was successfully conducted as lignin content in the EFB reduced significantly. However, it was only conducted in a small-scale production and not tested on animals yet. Following that, the purpose of my study was then to firstly replicate the production of EFB in an upscale amount, secondly to evaluate the intake, digestibility and rumen fermentation characteristics of the treated EFB as a total mix ration in a feeding trial. So EFB fibers from palm oil mills were pre-treated with mycelia culture of Ganoderma lucidum for 12 weeks of solid-state fermentation. Then the product was dried and shredded and made into a total mixturation diet of 3 different treatments, which was used in a feeding trial. Samples were then collected during the feeding trial for these 7 analyses and analyzed using statistical analysis system. Next, the results of the dry matter intake, fecal output, rumen pH, ammonia nitrogen, and volatile fatty acids were not significantly affected by the different treatments and were comparable to the control. Other than that, the apparent nutrient digestibility was also not affected except for the lignin digestibility or ADLD as seen in this graph. The ADLD of treatment 2 was lower compared to the other treatments, and this was due to the higher lignin content of the untreated EFB used in the treatment as compared to treatment 3, which contained pre-treated EFB instead. In addition, higher lignin content causes a lower fermentation and digestibility rate in the rumen, and this caused the feed consumed to have a slower disappearance through the digestive tract, which limits feed intake. As for the microbial population, total protozoa was not affected unlike the total bacterial count. So treatment 2, which contained untreated EFB, had the lowest bacterial count, and it was postulated that this was a result of high lignin content in the untreated EFB, since lignin is a complex polymer that is hard for bacteria to digest, which this then affects the availability of structural carbohydrates for bacterial growth. And since treatment 3 had a higher bacterial count than treatment 2, this indicated that the biological pretreatment applied on EFB provided better growth and improved fermentation activities of the bacteria. To conclude this study, the biological pretreatment using Ganoderma lucidum on EFB in a larger scale was successful as it has proved to effectively degrade lignocellulosic materials compared to untreated EFB. And when formulated in a total mix ration at 25% inclusion, it improved the lignin digestibility and supported the rumen fermentation activities comparable to the control treatment, making it suitable as feed for goats. So before I end my presentation, I would like to thank University Malaysia Sabah for this research opportunity given, Jabatan Perkhidmatan Veterina Sabah for providing my on-field research needs, IOI and PPB Group for supplying me the EFB source. These are my references in this presentation, and that is all from me. Thank you.